The Sam and Rose Stein Institute for Research on Aging is committed to advancing lifelong health and well-being through research, professional training, patient care, and community service. As a nonprofit organization at the University of California, San Diego School of Medicine, our research and educational outreach activities are made possible by the generosity of private donors. It is our vision that successful aging will be an achievable goal for everyone. To learn more, please visit our website at aging.ucsd.edu.
Wonderful. Well, I'm honored to be here, and I really want to thank the Stein Institute for the kind invitation and for the opportunity to present my novel research findings to you. The title of my presentation is Slowing the Clock, Longevity Science Meets Alzheimer's Prevention, and I'm Associate Professor of Public Health and Medicine at UCSD. So first I want to talk about the heterogeneity of aging. So we all age at different rates with diversity and functional decline, despite being of the same chronological age. So, there are people who age who have good mobility at age 90. There are others who have poor mobility at age 90. You have some people in walkers, some people, who are very vigorous and are able to engage in many activities when they're older. And aging is the strongest risk factor for common chronic conditions and disability that impact older people. But what we are learning in the past several years is that aging is, in fact, a modifiable risk factor. And what I mean by that is that the biology of aging is a modifiable risk factor. So we are gaining a better understanding of how we could potentially modify our biology of aging to prevent chronic disease and disability in older adults. And I will talk about that in the next slide. I'd like to introduce to you a topic called biological age. So we have chronological age and then we have biological age. So chronological age is calculated simply from your date of birth. So your age 60 or your age 80 or your age 90 or 95, etc. But biological age is a different concept. So biological age is the age-related decline in physiological functions and tissues. From biological age, we can essentially determine how fast your cells are aging. And we can determine whether you have what's called accelerated biological aging. So accelerated biological aging means that you are older biologically relative to your actual age. As an example, you might be a 60-year-old man, but your cells might be like 80 years old. On the other hand, you might be 60, but your cells are age 45. And why that is important is that through emerging research, we are beginning to appreciate that people whose biological age is older than their chronological age may be at heightened risk of chronic disease and disability. And what we are learning is that biological aging is a strong risk factor for multiple chronic conditions and disabilities that impact older adults. And if we could slow biological aging one day, then we could potentially reduce the risk of multiple chronic conditions and disabilities at once. There are many different ways to measure biological aging, or essentially the age of your cells. One approach are epigenetic clocks, and some of you may have heard of this or have had this measured in the past. So epigenetic clocks, so we take a drop of blood, for example, and from that we take your DNA, and within your DNA, there are specific chemical modifications that are reversible. And the combination of those chemical modifications can tell us how old you are biologically. The picture on the right shows us how epigenetic clocks work. From an epigenetic clock, which is measured in your blood using your DNA, we can determine whether you're biologically older or biologically younger. So this picture shows an individual at the age of 60. If he is biologically older, meaning that his cells are aging faster relative to his actual age, then he's shown to be here in a wheelchair and he's disabled and he has poor physical functioning. On the other hand, if he's biologically younger, meaning that his cells are younger than his actual age, then he's shown to have aged better and to be vigorous. The reason why this is important is because emerging research, specifically focused on epigenetic clocks, is showing that people who are biologically older relative to their actual age may be at heightened risk of chronic disease and disability. I was part of a collaborative group with researchers at Yale and others. We developed organ-based epigenetic clocks, and we published this in the journal Nature Aging last year. So through organ-based epigenetic clocks, from a single drop of blood, we can measure how fast your various body organs are aging. In other words, we can measure from a single drop of blood the biological age of your various body organs. So we developed epigenetic clocks for the heart, the lung, the kidney, liver, brain, immune system, an inflammatory clock, a blood clock, a musculoskeletal clock, a hormone clock, and a metabolic clock. We developed these clocks by training your epigenome using artificial intelligence to identify specific chemical modifications in your DNA whose combination can tell us how fast your various body organs are aging. And the reason this is important is because through these organ-based clocks, we can not only measure what your biological age is in various body organs, but we can predict how you will age into the future and whether you will get chronic disease and disability using these organ-based clocks. So the next step for us was we wanted to see whether these organ-based clocks were related to cognitive and brain outcomes, specifically Alzheimer's disease and related dementias. So I have been involved in a study for a long time called the Women's Health Initiative. The Women's Health Initiative is the longest study of women's health that has ever been conducted. It has been going on for over 30 years. And I was funded to measure epigenetics in 6,000 women from the Women's Health Initiative memory study to determine whether epigenetic clocks relate to Alzheimer's disease and related dementias. So we measured the organ-based epigenetic clocks when women were ages 65 to 79 using stored blood specimens, and women were followed for 25 years to determine incident mild cognitive impairment and dementia. And here are our main findings. We found that women who had accelerated blood aging, meaning that their blood cells were older biologically relative to their actual age, were at higher risk of incident mild cognitive impairment and dementia by 9%. We also found that women who had accelerated heart aging, meaning that their hearts were aging faster biologically relative to their actual age, had higher risk of mild cognitive impairment and dementia by 9%. And we also found that women who had faster metabolic aging also had heightened risk of dementia by 12%. The other organ-based clocks were not related to mild cognitive impairment or dementia. Okay, so we next studied plasma Alzheimer's biomarkers. So some of you may have heard of these. So in the past several years, there have been significant advances in identifying blood-based biomarkers that can tell us whether there's Alzheimer's pathology in your brains. There are many of these biomarkers that have been developed. These include p-tau217 and p-tau181. So those are two blood-based biomarkers that can especially p-tau217, can strongly tell us and very robustly tell us whether you have Alzheimer's pathology in your brain, meaning that whether there are changes in your brain that suggest that you have Alzheimer's disease. Other biomarkers in the blood that have been identified are amyloid beta 42 and 40, which indicates amyloid plaque buildup, although that is not as sensitive as p-tau217. Others include neurofilament light, which is a general marker of neurodegeneration, and leofibrillary acidic protein, which is a marker of inflammation in your brain. And the reason these blood-based biomarkers hold such promise is because they can help us detect Alzheimer's early. So they can potentially help us one day detect Alzheimer's several years before symptoms develop. They reduce the need for invasive testing using cerebrospinal fluid or invasive PET scanning. They can help us track disease progression, and these blood-based biomarkers of Alzheimer's disease are being increasingly used to recruit patients into clinical trials. But p-tau217 is the most promising biomarker thus far. So what we sought to do within my specific study, the Women's Health Initiative Memory Study, we wanted to see whether these organ-based clocks, which measure biological aging in various body organs, were related to these plasma biomarkers of Alzheimer's disease. And what we found was that, and so we correlated the organ-based clocks to changes in the plasma biomarkers of Alzheimer's disease over 15 years follow-up. And what we found was that women who had faster aging of the heart, meaning that their heart was older biologically relative to their actual age, they were more likely to show increases in p-tau217, GFAP, NFL, and p-tau181, indicating that faster aging of the heart was associated with Alzheimer's disease progression. We also studied whether faster aging of the blood, so whether people who had blood cells that were older biologically relative to their actual age had faster increases in p-tau217 as well as faster increases in GFAP, which is a marker of neuroinflammation, but had no change in NFL or p-tau181. So really what we found here with these analyses was that these organ clocks, which tell us how fast your various body organs are aging, are also correlated with incident mild cognitive impairment and dementia, really focused on clinical symptoms, and also were correlated with Alzheimer's disease pathology in your brain as measured by these blood-based biomarkers. So the take-home message from this analysis is people whose heart and blood was older than their actual age, they were more likely to have higher risk of cognitive impairment and experience progression of Alzheimer's disease as indicated by these blood-based biomarkers. And those of you who may ask, do these findings apply to men? We did not study men in our specific study. However, there are emerging studies that are showing that organ-based clocks also predict incident cognitive impairment in men as well. There are some studies that have shown that so far. Okay. Within the Women's Health Initiative memory study, we next wanted to examine whether plasma p-tau217 predicted incident mild cognitive impairment and dementia. So we measured plasma p-tau217, which again is a blood-based biomarker that can detect changes in your brain indicating Alzheimer's disease. We measured this biomarker at baseline when women were cognitively healthy and they had no cognitive impairment or dementia. And we wanted to determine whether this blood-based biomarker could predict the incidence of Alzheimer's disease and related dementias up to 25 years later. And what we found was that women who had higher levels of plasma p-tau217 had a 143% higher risk of mild cognitive impairment and dementia up to 25 years later. So these women, remember, had no cognitive symptoms at baseline, and this blood-based biomarker could predict whether they are at higher risk of mild cognitive impairment and dementia up to 25 years later. We next want to see whether there are key factors influencing the risk. And what we found was that the findings were stronger in women who were older, in women who had genetic risk for Alzheimer's, specifically carrying the ApoE4 gene, which indicates strong genetic risk for Alzheimer's disease. And we also found that the association between plasma p-tau217 and incident mild cognitive impairment and dementia was strongest in women who were on hormone therapy, specifically on estrogen plus progestin therapy. So the take-home message from this analysis was that plasma p-tau217 may be a robust biomarker measured in the blood that can predict future mild cognitive impairment and dementia decades in advance among older women. And I just also want to add that plasma p-tau217 in other studies has also been shown to predict dementia robustly in men as well. And the findings from this study will be published in the journal JAMA Network Open. It was recently accepted for publication, and it'll be published in about 6 weeks. So traditionally, these plasma biomarkers of Alzheimer's disease, have been studied in relation to primarily cognitive and brain outcomes. We were interested in studying these blood-based biomarkers in relation to aging outcomes, specifically exceptional longevity and health span. So we wanted to see whether these plasma Alzheimer's biomarkers might also have utility outside of Alzheimer's disease. So what we did was we wanted to see whether these plasma Alzheimer's biomarkers predicted whether women would live to age 90 or older. And what we found was that many of them did predict exceptional longevity. So specifically, we found that women who had higher levels of plasma p-tau217 had a 30% reduced odds of living to age 90 versus death. We also found that higher levels of p-tau181 reduced the chances of living to age 90 by 11%. NFL was also robust. It reduced the chances of living to age 90 by 25%. And remember, NFL is a general marker of neurodegeneration. So this was interesting, and death due to dementia did not explain our findings. So this tells us that these blood-based biomarkers of Alzheimer's disease might have utility outside of just predicting cognitive and brain outcomes, and might have utility as potential biomarkers of aging. We also wanted to see whether these plasma Alzheimer's biomarkers predicted whether women were healthy at age 90. So we defined healthy at age 90 as women who survive to age 90 without mild cognitive impairment or dementia. And what we found was that many of these biomarkers did robustly predict healthy aging at age 90. Specifically, higher levels of plasma p-tau217 lower the chances of healthy aging by 42%, indicating how important this biomarker is to not only predicting cognitive and brain outcomes, but also longevity and healthy aging. The other biomarkers also were predictive, especially NFL, which is the general marker of neurodegeneration. The take-home message from these analyses that we did was that plasma p-tau217, specific and plasma biomarkers in general, may not only be useful to predict future risk of dementia, but they also may be useful to predict aging outcomes that are of great interest to the aging population, specifically longevity and healthy aging, because an older adult will want to know, will I age well into the future? And if I'm predicted not to age well into the future, what can I do about that now to change that trajectory? So that's why blood-based biomarkers and blood-based biomarkers of aging are so important to predict the future, whether you are expected to age well into the future, and if you're not, based on your current biomarker levels, could we possibly do something about that? So the next question you may ask is, well, what are some modifiable risk factors that might be associated with changes in these plasma biomarkers of Alzheimer's disease? So we did an analysis where we wanted to see whether whether cardiovascular risk factors predicted changes in the blood-based biomarkers of Alzheimer's disease. And what we found was that women who had diabetes, women who had long sleep duration, specifically who slept more than 8 hours per night, women who had hypertension, obesity, or smoking generally had higher levels of many of these biomarkers. We also found that past alcohol consumption was protective. So we found that women who reported consuming alcohol, they had slower increase in GFAP, which was the marker of brain inflammation. So I also want to add that many of these biomarkers, many of these cardiovascular risk factors have also been associated with changes in these plasma biomarkers in men as well. So the take-home message from this analysis was that, how important, well, we know in general that cardiovascular risk factors increase risk of dementia, but what we are showing here for the first time is that cardiovascular risk factors may also be associated with reducing the progression of these plasma biomarkers of dementia pathology, which which detect the pathology of dementia in your brain. So targeting and controlling cardiovascular risk factors is very important. Okay, so I'd next like to talk to you about one of my other studies that I've been involved with the last few years. So we have been doing what's called proteomics, the study of proteomics. So proteomics is a study of proteins, essentially. And we received funding to use this cool new technology called SomaScan to measure proteomics within the Women's Health Initiative memory study. So the SomaScan is a cool technology that simultaneously measures 11,000 proteins in your blood. It has been leveraged to discover novel proteins and biomarkers for chronic diseases and aging outcomes. And because it has such broad coverage, it can be used to identify novel pathways for disease and disability. It's reproducible, it's very specific, and it's very sensitive, and it's a very cool new platform to study proteomics in chronic disease and disability. So one of my collaborators at Wake Forest University, Ramon Casanova, he leveraged the SomaScan platform to see whether proteins measured in SomaScan could improve 10-year prediction of dementia. So Ramon applied machine learning an artificial intelligence to this large data set of of nearly 3,000 women who had 11,000 proteins measured in their blood, and he wanted to see whether the combination of specific proteins with age and plasma biomarkers of Alzheimer's disease could improve prediction of dementia. And here's what we found. So first, he did an analysis looking at the combination of SomaScan proteins and age. And the combination of that resulted in a C index of 0.73. So a perfect C index indicating a high accuracy is 1. So the closer to 1, the higher the accuracy. When he combined only blood-based biomarkers of Alzheimer's disease and age, the C index was 0.81. But when he combined proteins from SomaScan, plasma biomarkers of Alzheimer's disease and age, the C index increased to 0.84. That was the best performing machine learning model, indicating to us the promise of combining demographic factors, blood-based biomarkers of Alzheimer's disease, and proteomics to improve 10-year prediction of dementia. I also wanted to add that while we studied women in this specific population, in this specific study rather, other studies have also shown that proteins and combined with blood-based biomarkers of Alzheimer's disease and age also improve prediction of dementia in men as well. Okay, so earlier in this presentation, I talked to you about organ clocks measured using epigenetics, or organ clocks measured using chemical modifications in our DNA. We can also measure organ clocks, or how fast your various body organs are aging, using proteins in your blood. So the SomaScan platform, which again measures 11,000 proteins in your blood at once, the SomaScan platform was leveraged by researchers at Stanford to to identify novel protein organ clocks of aging, specifically in the heart, the liver, the muscle, kidney, artery, immune system, blood plasma, and adipose tissue. And these are developed by applying artificial intelligence models to proteins in the blood to identify combinations of proteins that can tell us how fast your various body organs are aging. So we want to see whether specific proteomic protein organ clocks were associated with mild cognitive impairment and dementia and healthy aging within our study in the Women's Health Initiative. So here's what we found. This shows the associations of many different protein organ clocks with mild cognitive impairment and dementia in the Women's Health Initiative memory study. So what we found was that the brain clock, the heart clock, the immune system clock, and the pancreas and liver clocks all were associated with incident mild cognitive impairment and dementia. Meaning that, for example, women whose heart was biologically older relative to their actual age had a 14% increased risk of mild cognitive impairment and dementia. Women whose immune system was older than their biological age had an 11% increased risk of mild cognitive impairment and dementia. So the heart clock was the most strongly associated with mild cognitive impairment and dementia in this specific analysis. We also want to see whether these protein organ clocks predicted healthy aging. We defined healthy aging as survival to age 90 without mild cognitive impairment or dementia. So we found that faster aging of the artery, the brain, the heart, and the immune system lower the chances of achieving healthy aging. So essentially, having accelerated biological aging in these specific body organs, like the artery, the brain, the heart, and then also the cells of the immune system, all lower the chances of achieving healthy aging. And I think why these types of analyses are so important is that we don't really fully understand why some people reach age 90 and older without chronic disease or disability. There are many people who are, supercentenarians or semi-supercentenarians and nonagenarians who don't have chronic disease or disability. And through these types of analyses that we're doing with these protein organ clocks, we are understanding why. And one of those reasons might be that these people who age 90 and older without, in this specific example, cognitive impairment or dementia, have slower biological aging in those specific body organs. So biological aging might play an important role in healthy aging. This just shows additional clocks that predicted healthy aging. So the kidney clock was in fact the strongest. So people whose kidney was older than was older biologically relative to their actual age, they had a 30% lower chance of achieving healthy aging. People whose liver and also the liver and pancreas clocks also predicted healthy aging. So the take-home message from this analysis was that the heart clocks and the kidney clocks, they were most strongly associated with dementia or healthy aging, indicating how important the biological aging in these body organs might be in Healthspan. Okay, so I've talked to you a lot about how biological aging relates to healthy aging and mild cognitive impairment and dementia. So the next natural question is, well, what can we do about it? How can we slow biological aging? What can we do to increase our health span or years of life lived healthy? So I'd like to spend, the next few slides talking about that. So there are many types of interventions that are currently being tested to see whether they can increase your health span. So by health span, I mean the years of life lived healthy. People are testing lifestyle interventions, including physical activity interventions, dietary interventions, weight loss interventions. There are many companies and startups that are trying to identify novel medications to increase health span. Now, something we should add is that aging is not yet recognized as an indication by the Food and Drug Administration. So we're not at the point of where we can say we have a drug that necessarily targets aging, but we will be there in the future. There are also many people who are trying to identify existing medications that can be repurposed to increase your health span, which I'll talk about in a later slide. And there are other companies that are testing cell replacement therapies, gene therapies, immunotherapies, etc. But I just wanted you all to appreciate that there are many people who are trying to identify novel interventions that can help us slow biological aging and achieve healthy aging. And one of the best ways to achieve healthy aging is through exercise. Exercise is the best medicine. And I like to talk about a clinical trial that I was involved with called the EXERT Trial. So the EXERT Trial was a collaboration between UCSD and Wake Forest University, which is a trial among older adults with mild cognitive impairment. We want to see whether exercise could slow cognitive decline in older adults with mild cognitive impairment. The findings from our trial were published a few months ago in the journal Alzheimer's and Dementia, and I'll talk to you about these findings in the next few slides. So the EXERT trial, it recruited 296 men and women, 86% white, 10% black, average age of 74 years, all had mild cognitive impairment, so they had some memory and thinking problems, putting them at heightened risk of future dementia. And these men and women were randomized into two arms in the trial, an aerobic exercise arm, where they participated in aerobic exercise 4 times per week with a trainer at the YMCA for 30 to 40 minutes per session. They did things like the elliptical or the treadmill, for example. And the other arm was a light intensity activity arm. So they did stretching, balance, and range of motion exercises 4 times per week with a trainer, 30 to 40 minutes per session. They did this for about 12 months, supervised with a trainer at the YMCA, and then 6 months continued with that level of activity without trainer supervision. So we measured changes in cognition in these men and women over the 1 year follow-up. We used a measure called the ADAS-Cog-Exec, which is a measure of general global cognitive functioning. And we did not find any significant difference between the high intensity exercise group versus the low intensity exercise group. But a very interesting finding to emerge from our analysis was that neither group experienced cognitive decline over the 1 year follow-up. And that was surprising because we had anticipated that they would, because they were older adults with mild cognitive impairment, and we anticipated that they would show some decline over the 1 year follow-up, but they didn't. So our next question was, in our original analysis, we compared 2 exercise groups together, but we next want to see whether if we compared each exercise arm to a usual care group. So a usual care group was a group that did not participate in any exercise at all. So we want to see whether participation in any exercise improved cognitive functioning. And what we found was that the usual care group, so that was the group of people who did not do any exercise at all, they experienced cognitive decline over the 1 year follow-up, but the aerobic exercise group did not. So when compared to a group of people who did no exercise at all, participation in aerobic exercise resulted in less cognitive decline. We then compared the usual care group, which is again the no exercise group, to the stretching, balance, and range of motion group. So that was the low intensity exercise group. And we also found similarly in this group, the usual care group got worse over 12 months, but the light intensity group did not. So in summary, what we saw was that when we compared the 2 exercise groups together, there was no difference. Neither group experienced decline at all. But when we compared each of the arms to a group that did no exercise at all, that's where we saw a difference. So the take-home message from this analysis was that exercise of any intensity, whether it was aerobic or low intensity, like doing stretching exercises, may slow cognitive decline in older adults with mild cognitive impairment. And I think something I also want to mention is that in older adults, doing light activities has been shown to improve health outcomes. And light activities include things like doing chores around the house, or even doing things like doing gardening. So avoiding sitting too much and doing some activity, even if it's light walking, chores in the house, gardening, that all counts as light activity. So I just wanted you to remember that as you think about, these findings. And the key was to target exercise of any intensity for 120 to 150 minutes per week. Okay, so I talked to you about a lifestyle intervention, which is which is exercise, but I also like to talk to you about a drug called metformin. So metformin is a medication that is taken by people who have type 2 diabetes. And metformin has been shown to improve biomarkers of aging. So there's been some evidence indicating that metformin might be a medication that potentially could slow biological aging and prevent multiple chronic diseases and disabilities in older adults, although we need a robust clinical trial to help us get at that. We wanted to see whether metformin was associated with exceptional longevity, specifically survival to age 90. So we did an analysis within the Women's Health Initiative where we compared women who were on metformin to women who took sulfonylureas. So sulfonylureas are another type of diabetes medication. And we want to see whether metformin resulted in longevity. And what we found was that women who took metformin had a 30% lower risk of death before age 90 compared to women who took sulfonylureas. So in our specific study, our findings suggest that metformin when compared to sulfonylureas, increase exceptional longevity. There have also been other studies that have shown that metformin reduces risk of death in men as well. So this is a drug that has been shown in both men and women to, or in some studies, not all, to reduce mortality. And the findings from this study were published in the Journal of Gerontology last year. The take-home message from this is that, metformin might be a medication that could potentially slow biological aging based on prior evidence. And we show for the first time that it could also potentially increase a person's length of life, specifically their survival to age 90. In conclusion, I have three main conclusions from my presentation that I hope you take with you. I hope you learn more about this concept of biological aging, how if you're older biologically in specific body organs, that might put you at higher risk of chronic disease and disability, specifically mild cognitive impairment and dementia, and reduce your chances of achieving healthy aging. But I also don't want you to think that, that's not something you can reverse. So what the important public health message from this is that, biological aging is something that can be reversed, and that's what we're learning. And that's the beauty of it, is that it's a modifiable risk factor that we want to target either through lifestyle interventions or through medications one day to slow biological aging. Because if we could slow biological aging, then we could potentially prevent multiple chronic diseases and disabilities in older adults at once. Another important finding from our analyses was that these very exciting plasma biomarkers of Alzheimer's disease, like p-tau217, p-tau181, they may not only have a utility for predicting cognitive and brain outcomes, but our findings suggest that they could potentially be useful to predict how long you will live and whether you will live healthy. So they might also have utility as potential biomarkers of aging. And finally, the EXERT trial, as a randomized clinical trial, it provides very strong evidence how important exercise may be to slow cognitive decline in older adults who are having memory and thinking problems. With respect to future directions, so I have been studying exceptional longevity, healthy aging, Alzheimer's disease for a very long time, and I'm currently seeking funding to do a study of healthy aging in centenarians. This is very personal to me because my grandfather lived to the age of 102, and he lived to the age of 102 very healthy. And so, within the Women's Health Initiative, we have data on women who were followed from ages 50 to 79 to ages 90 and older, and we now have data on over 2,000 centenarians with over 30 years of follow-up data, which would allow us to determine what are the determinants throughout the life course that allow women to reach age 100 years and older in good health. And so I'm currently seeking funding for that. Haven't been able to receive it yet, but I'm persevering, and I'm really hoping to receive funding for that to be able to do this important study with the data we have. I would like to conclude by thanking everyone who made this research possible. I would like to thank all my collaborators at UCSD, as well as the leadership in the School of Public Health. I would like to thank the Stein Institute for their kind invitation and also for all the hard work they put into promoting this event. I would like to thank my collaborators at Wake Forest University, at the National Institute on Aging, at Harvard, NYU, University of Washington, Altos Labs, Brigham and Women's Hospital, and the University of Minnesota. As you can see, the work that we were able to do was very collaborative, and this work would not have been possible without the expertise and the collaboration of all of my collaborators who are listed here. And very importantly, I would like to thank the 161,808 participants of the Women's Health Initiative, some of whom are pictured here, who made this work possible, who since 1993 have contributed their time to this landmark study of women's health that is currently ongoing, and that has allowed us to better understand the determinants of healthy aging and exceptional longevity. So here's my contact information, including my email, my cell phone. I'm also on Instagram and TikTok, so you can follow me. Screenshot this and feel free to contact me at any time. Again, I'd like to thank all of you who took the time to watch my presentation today, and I look forward to taking your questions.
Fantastic. What a great talk today, Dr. Shadyab. We have a lot of questions for you. I'd love to just go ahead and get started. First question, is epigenetic clock testing available to the general public, and how could we get our blood tested?
Yes, there are some companies that I believe measure that. I've never had it measured myself personally, but there are some companies that do measure that.
Okay, great. Okay. How can we have lower biological age? Are there any practices or treatments that can slow the clock?
That's another good question, and that's something that we're, still learning. What can really slow biological aging? Um, I think there have been some other studies showing that modifiable lifestyle behaviors like physical activity are associated with biological aging. There's been some recent studies showing that how diet is associated with biological aging. So there was a big landmark study that showed how caloric restriction might slow biological aging. So yes, there are lifestyle behaviors that might modify that. Metformin is another medication that has been shown to potentially slow biological aging. But I think that, we're still in the process of really trying to better understand what biological aging is, what it relates to with respect to disease and disability, and what we can do to slow it. So I think this field that we're in biological aging is still in its infancy, and there's still a lot left to learn.
That's helpful. Thank you. Okay, why do you think that sleeping more than 8 hours per night is a risk factor?
That's a very good question. And actually, when I was a PhD student, one of my earliest papers was how short and long sleep duration relate to type 2 diabetes. So in the literature, it has been shown that both short and long sleep duration are associated with poor health outcomes. And that includes many poor health outcomes like diabetes, cardiovascular disease, and even dementia. So the recommended average hours of sleep that a person should be getting per night is about 7 to 8 hours. So anything below or above that puts you in the short and long sleep duration categories. So this is something that a lot of research has shown. With respect to the mechanisms, I'm not fully. I think it just depends it depends on the specific disease that you're studying. But short and long sleep duration both have been associated with poor health outcomes in older adults.
Okay, great. So tell us about this one. Should we all test for our p-tau 217, or is there a specific group that should take that test?
Yeah, and again, I think that's something that so I know that FDA just approved a p-tau 217 test, and it's for people who have cognitive symptoms exhibiting Alzheimer's disease. So the p-tau 217 test is not at this time recommended for people who are cognitively healthy. The research that we're doing is getting at that. Does p-tau 217 predict, dementia into the future? We are not yet at the point of saying, oh, you should test for p-tau 217. There are many ethical implications in that, and it could actually potentially cause harm if everybody tested for p-tau 217. So we're not the point yet in the field, but it has been approved by the FDA, I believe, to for testing in people who have cognitive symptoms that are suspected to be Alzheimer's disease to reduce the need for additional invasive testing.
Okay. Someone's asking you to define the difference between MCI, dementia, and Alzheimer's.
Yeah, so within the women's health and so Alzheimer's is a subtype of dementia. Um, in mild cognitive impairment is an intermediate stage. So in mild cognitive impairment is when they have memory and thinking problems, but it's not full-blown dementia yet. So mild cognitive impairment it's a good time to intervene because the older adult will have memory and thinking problems, but it has not impacted their function enough yet to be full-blown dementia yet. So I want to go back to the EXERT trial because the EXERT trial was in older adults with mild cognitive impairment. So they did not have dementia yet. They had only memory, for example, memory and thinking problems. So we want to intervene through an exercise intervention to slow their cognitive decline and potentially, prevent dementia, although we did not look at dementia in that specific trial.
Okay, great. We have a couple of questions that have come in on estrogen and hormone replacement therapy and dementia. Um, any thoughts on that? Recent news indicated HRT can prevent dementia, but your findings show otherwise. Can you speak on that?
Yeah, and I appreciate that question. So far, the best evidence we have on the effects of hormone therapy on dementia are from the Women's Health Initiative Memory Study. The Women's Health Initiative Memory Study was a randomized clinical trial of hormone therapy. in postmenopausal women, which showed that uh, hormone therapy, whether estrogen alone or estrogen plus progestin, actually increased the risk of mild cognitive impairment and dementia. Now, I did want to add some caveats around that. Uh, it was among postmenopausal women, so women who were 65 years and older, so it does not apply to women who were younger or who took hormone therapy around the time of menopause. It also was around the time of use older hormone therapy formulations, and it's not may not apply to the current hormone therapy formulations. And so, yes, there are caveats around that, but having said that, it thus far remains the most rigorous, largest, and robust clinical trial, originally among 7,000 women,
Amazing.
that showed that hormone therapy increased the risk of mild cognitive impairment and dementia. And so that's why our study on plasma p-tau217 and incident mild cognitive impairment and dementia was so important, because we very rigorously got to see whether randomization to hormone therapy influenced the associations of plasma p-tau217 with these outcomes, which no other study is able to do because they rely upon self-reported hormone therapy.
Okay, fantastic. Okay. Does blood type have any effect on the risk of dementia and or longevity?
I don't know. I haven't studied that. So I can't answer that question, but I can look it up and get back to you.
Okay. How about this one? What about nutritional modifications to help reverse Alzheimer's dementia? Any literature on that?
Yeah, I know there was some large there was, oh well, yeah, actually, so the US Pointer trial. So I did not talk about the US Pointer trial. But that's a trial you want to look up, because I believe that trial it targeted diet, exercise, and other things and it had positive findings for affecting cognition. Now, I'm not fully familiar with the U.S. Pointer trial, but that's one trial you want to look up. If you Google U.S. Pointer, you can see how a multimodal intervention had a very positive impact.
Okay, great. Okay, here's a tricky one. How do you assess if organs are older than actual age?
Yeah, so that's getting mathematical. So we essentially we measure the biological age, for example, from the proteins or from the epigenetic clocks, and then we regress that on age in a linear regression model, and then the residual from that model is essentially the measure that we use that tells us that we call, for example, age gap, which is the difference between biological age and chronological age. So we get it from a mathematical linear regression model. The residual from that model tells us the difference between biological age and chronological age. And so we're like, for example, positive values indicate that you have accelerated biological aging, and smaller values tell us you have slower biological aging.
Okay, and what about metformin? Does it slow down aging just for people who are diabetic, or are there benefits for the general public?
So far, the most of the evidence in metformin has been in people who have type 2 diabetes. For example, the data that we used was in people who have type 2 diabetes, because it's a medication that's given to people who have type 2 diabetes. Now, there has been great interest in starting a clinical trial that to give metformin to people who don't have diabetes, to see whether it slows aging, slows biomarkers of aging, and reduces risk of disease and disability. They were trying to get it off the ground. It's called the TAME trial. It hasn't started yet. I believe it was due to they had to get funding, and the funding didn't come through, I believe. So we don't know yet whether, from a clinical trial standpoint, whether metformin can, uh, you know, is helpful for people who don't have diabetes.
Okay. And then time for our last question. What are your thoughts or experiences with various forms of fasting, like intermittent fasting, and its effect on biological aging and dementia? Do you have thoughts on
Yeah,
that?
yeah. So I believe there is some evidence that that may have an effect. So I think I told you earlier, um, there was a study in Nature Aging that looked at caloric restriction and how that slowed biological aging. And so, yes, there is evidence of that. And so, yeah, so I think, in conclusion, I think there's a lot of promise when it comes to the study of biological aging, and how we could potentially target it through combination of lifestyle interventions and potentially medications to slow biological aging and prevent chronic disease and disability in older adults.
Okay. Well, Dr. Shadyab, what an amazing talk today. Thank you so much. The talk was fantastic. I hope all of you enjoyed it. And a reminder that all of our past newsletters, updates, ongoing research, training programs, and past UCTV lectures can all be found on our website at ucsd.edu. And if you are interested in supporting our fundraising campaign to allow this work to continue to go on, we would love to get your support there. You can either contact me or go to our website at aging.ucsd.edu, where there's a place to donate right there on site. So with that, I want to thank Dr. Shadyab for such an amazing talk, an amazing presentation, and thank you all for joining us today, and hope to see you again in 2 months.